Today, we will be visiting Target Malaria's insect tree in Entebbe, Uganda. Target Malaria's work in Uganda is led by the Uganda Virus Research Institute, UVRI, and the insect tree is within its premises. Target Malaria is an international not-for-profit research consortium working to develop and share new, cost-effective, and sustainable genetic technologies to modify mosquitoes and reduce malaria transmission. The project brings together several research institutions across Africa, Europe, and North America, including UVRI. The UVRI was established in 1936 and conducts research on communicable diseases, especially viral diseases. The institute has a department of entomology and vector biology, which focuses on the study of various arboviral vectors and is noted for the discovery of several mosquito-borne diseases. The institute's findings inform government strategies for vector control. The insect tree is a standalone, self-contained facility, which was first launched in July 2019. It is currently used to conduct analysis on wild-type Anopheles gambia mosquitoes, one of the main vectors of malaria in Africa. This work helps understand local mosquito population dynamics and inform modeling and genetic studies. Following regulatory approval, the insect tree will be used to carry out studies on genetically modified mosquito strains and support the field evaluation studies if the research progresses and these are approved.
Amanya gange nze Dr. Kato Agapitas.
Sanyise kusinkana ssebo nange amanya gange nze Mawanda Nasiri.
Mawanda Nasiri. Sanyise okulaba
hanyi.
Tusanyise okulaba mu sekitale yaffe eno.
Safety is paramount at Target Malaria. Before any visitor is given access to the insect tree, they must watch a short presentation that introduces them to the project and lays out the rules and guidelines to follow when visiting the insect tree. After watching this presentation, visitors must sign a waiver. They are then officially ready to enter the insect tree. The insect tree is equipped with a number of safety features to ensure regulatory compliance. The facility has restricted access and window openings are sealed. The vestibule, which is the only room leading into the ACL2 premises, is equipped with a double lock and airlock system, which ensures that no insects can get through it. There are static mosquito traps on the wall to ensure that any mosquito that gets to this point is trapped. Handheld electric mosquito traps are also available to serve as an extra verification that no mosquitoes are introduced or escape. This is the wash-up room. All wastewater from the ACL2 area drains through a special filtration system that prevents mosquito eggs, larvae, or pupae from being washed into the environment. The room is also fitted with an autoclave to take care of all solid waste and a washing machine to treat protective gear after use. An important part of the work we do in the insect tree is to maintain mosquito colonies to carry out research. This room currently houses a wild-type colony of mosquitoes. Younger mosquitoes, in form of pupae and larvae, live in trays whilst adult mosquitoes are kept in cages. The colony is maintained to allow researchers to carry out studies on mosquito rearing as well as to practice maintaining mosquitoes at ACL2 standards. Sorting of adult mosquitoes to differentiate between male and female mosquitoes is carried out in the identification room. The room is equipped with molecular analysis, DNA extraction, and DNA amplification tools in preparation for future research that will take place here. When the mosquitoes have reached the pupae stage, they are sorted by sex to separate males from females. This is done under a microscope. It is a highly labor-intensive process that requires a keen eye and a lot of practice from the scientists. Once sexed, the males and females are placed into cages to emerge to adults. The team carries out several tests on the mosquitoes present in the lab. One of these tests are PCR tests, through which researchers analyze the DNA of mosquitoes to determine their species.
So with the identification, we start with a process called extraction. We're trying to extract the DNA from the mosquitoes. So we use a couple of machines, including the heat block, the centrifuges, and the vortex mixers, plus the water bath. So after extracting this DNA from the mosquitoes, we want to amplify these segments that we talked about, the ones which will tell us which species the mosquito belongs to. So we mix that DNA with a couple of other reagents and we run it in what is called a thermocycler. So this is what runs the PCR, or the polymerase chain reaction. The process runs for about 2 to 4 hours depending on which protocol you're following. And after this, we now want to see which segments have been amplified. And these segments will tell us which species the mosquito belongs to. To do that, we can't view these segments with our naked eyes. So we have to run a process known as Gale electrophoresis, which is a process whereby the DNA, there's a special light we pass through that DNA, and it will fluoresce, or it will absorb the light and give off a specific kind of light. So when the light goes through that DNA, we can be able to know which specific segments have been amplified. But we can't use our eyes here, that's why we have a camera that captures the image, and the image is sent to the computer where we can do our analysis. So from there we can know that the mosquito we were dealing with belongs to species A or species B.
In order to maintain mosquito colonies for our research, the mosquitoes in the insectary are made to mate. We currently practice this with wild-type mosquitoes, but as our research progresses, we will be working to mate wild-type and genetically modified mosquitoes to create and maintain a strain of mosquitoes that carries a genetic modification.
Mating happens in the adult cages after sorting our pupae from the lavatories and sexing them into males and female. We transfer them to the adult cages. In the adult cages, we allow them about 4 to 6 days. In these 4 to 6 days, the pupae emerge into adults, and this period is again enough for them to begin mating. Mating is very important here in the insectary as it enables us propagate the next generation in the colony. During mating, male adults insert their sperm into the spermatheca of female adults, and this allows fertilization of the egg to take place in the female. What happens is in this way we are able to obtain viable eggs that are hatched and reared to give the next generation of the colony.
Mosquitoes also need to feed, and the team feeds the larvae on pellets suspended in water. Once they have grown into adults, they are maintained on a 10% sugar solution. In addition, female mosquitoes receive a blood meal to ensure reproduction. The team monitors environmental conditions in the insectary using a digital system. The system monitors temperature and humidity. The optimum temperatures for mosquito growth in the facility are 25 to 28 degrees centigrade, while relative humidity is maintained at 65 to 85%. Termination of mosquitoes is aimed at management of mosquito stocks in the facility. Surplus larvae and pupae are terminated by immersion into hot water, while aged adult mosquitoes in cages are terminated by spraying them with ethanol. These are then disposed of as solid waste in waste bins prior to autoclaving and incineration.
Haji,
Sebo,
ekifo kyaffe kyekyo nga bw'okirabye.
weewaawo Sebo, eyanziza nnyo.
Community interest in our work is always encouraged and allows us to remain accountable and transparent in our work. While we welcome visitors to the insectary, we need to manage the number of visits in order to minimize disturbance to the mosquitoes and to our team's work. We hope this video gave you a better understanding of our work and answered some of the questions you may have had. At Target Malaria, we are committed to being open and accountable as we work to develop an innovative tool for malaria control.